Today we're going to talk about an incredibly powerful tool, CRISPR, short for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPR is a tool that allows us to edit the very building blocks of our bodies, the genes. Genes are found in every cell and dictate how proteins are formed. Proteins make up almost everything in our bodies. CRISPR is able to take small slices out of our DNA, cutting out chunks with extreme precision. It's actually a natural process found in bacteria to protect against invading viruses. Let's take a look at this process and then the further applications in humans.
Here's a bacteria. It is a single-celled organism and there are billions of them in your body right now. Don't worry though, as most are either harmless or even helpful to your body. When a virus attempts to invade our bacteria here, an enzyme called Cas9 soars over to the virus and captures a piece of the virus's DNA. The Cas9 then takes this DNA to CRISPR and copies the DNA into this defense mechanism. Now, whenever the virus, or a similar one, attempts to invade, it stands no chance against the mighty CRISPR. CRISPR in humans is very similar. There is the Cas9 enzyme as well as a nucleotide RNA. This just helps lead the Cas9 to the exact location. The enzyme then proceeds to cut out the pairs that it needs to. After that, the DNA is fragmented with two blunt edges. There are two different repair methods. The first, non-homologous enjoining, occurs naturally in cells. The DNA is repaired with a band-aid of sorts, and the two edges are patched together haphazardly. To do this, sometimes pairs are removed, turning the gene completely off. The second way, homology-directed repair, allows scientists to input whatever pairs they wish. They insert a copy of the genome with no differences besides what they wish to change. This fools the DNA into incorporating the base pairs that scientists want to replace with. This allows the gene to stay healthy and functional.